Hello everyone, welcome back to my YouTube channel. Today videos is about the NK cell receptor and cell cytolysis. So we first know the what is the receptor. Receptor is a molecular structure, usually a protein, that is located on the surface of the cell or inside of the cell. So receptor have a specific binding site that allow them to interact with the particular molecules called the ligands. Okay, so receptor are made up of the protein. However, ligands can be various substances, including hormones, neurotransmitter, antigen, or other signaling molecules. So this is the structure of NK cell. Okay, so NK cell having number of receptor. This is the cytokine receptor, activating receptor. Here it is the both activating and inhibitory receptor. This is NKG2 and KIR. Inhibitory receptors. Okay, so this is all the NK cell receptor. Okay, so now we understand the NK cell receptor in two different animals like human and mouse. Okay, so you can see here this picture: activating receptor, ligand, inhibitory receptor, chemokine, cytokine receptor, adhesion receptor. Okay, so for example, activating receptor in human that also differ from the mouse. Okay, this all are the different from the human to the mouse. Now the human NK cell. On the other hand, human NK cells don't have the NK 1.1 but express CD56, which can vary in the density. Okay, we will discuss in later. Okay, so first we discuss the NK cell receptor or marker on mouse. Okay, most mouse NK cell can be identified by the presence of certain markers on their surface. Okay, so these markers include CD122, which is the subunit of the interleukin 2 receptor, and another that is the NK1.1, part of the NKR P1 family, and another that is the CD49B integrin. Here it is the okay, all are the mouse receptor, NK cell receptor. Now, additionally, NK cell typically have the CD2 and Fc gamma R2 on their surface, also having. Now, interestingly, if you use specific antibody against the Fc gamma R3, okay, so it can remove almost all the NK cell from the bloodstream. Now we know the human NK cell markers and receptor. Or receptor, we can say the so NK cell also express the interleukin 2 receptor and Fc gamma R3, but they don't have the NK1. Okay, it is 2 receptor. Okay, now instead of this, the human NK cell are identified by the presence of CD56. Okay, there is a 2 subunit of NK cells based on the density of the CD56. So CD56 bright, that is cytolytic produce cytokine, okay, and CD56 dim cell killing activity. Now the CD56 bright NK cells are similar to the T helper cells. Okay. So CD56 bright, okay, so bright cell consisting the majority of the NK cell being found in bone marrow, second lymph node tissue, liver and skin. Okay, so this is the human NK cells, the receptor of. Now here it is the CD56 bright NK cell, CD56 dim NK cell, and tissue resident NK cell and adaptive NK cells. Okay, so these are all the markers. Okay? CD56 CCR7, rare subset in the blood but abundant in certain tissue, as I said earlier, can rapidly produce immunomodulatory cytokine. Okay, so here it is the domain subset in the peripheral blood. Cytotoxic effector, okay, dim the role of the cytotoxic effector by releasing cytolytic granules, for example perforin and grain cell, which I have discussed in my previous video. Okay, and this link given into the description box, so please watch it. Okay, so now heteroresident NK cells Okay, population of cell found across various tissue. Cytokine and KIR profile. Okay, KIR, that is the another receptor, inhibitory receptor present in the human cells, differ from the conventional NK cells. Okay, they different from the conventional NK cell. Now, long-lived NK cell that can rapidly proliferate and produce cytokinin upon viral re-exposure. Okay, so this is the all markers. So here it is the picture. You can understand the how CD56 and CD56 dim and CD56 bright, they proceed their activity. Okay, and they leads to the cell cytolysis. So the first, virus NK cell activation. Okay, when NK cell is activated, when the virus infected cell release of the interferon alpha, beta and interleukin 2, 12, 15 and 18, which respectively stimulate the NK cell cytotoxicity and adaptive immunity activation. Now the first, CD56 dim. Here it is the CD56 dim NK cell has cytotoxicity function that can be mediated by the granzyme B production. Okay, in my previous video I have described in details that how the granzyme B is leads to the apoptosis. Okay, so and another, that is the FS and FSL pathway. This is also described in my previous video, so please watch it. And another, that is a CD16. Okay, CD16 and binding with the FC. Here it is the constant fraction, okay, of the antibody. Now adaptive immunity modulation is mediated by the CD56 bright. Here it is the CD56 bright. NK cell through the secretion of interferon gamma and tumor necrosis factor alpha that affect the T and B cells, and it is the granzyme B inserted into the infected cell and leads to the apoptosis. Now NK cell can eliminate virus infected cell via this CD60. Here it is mediated ADCC. Okay, antibody dependent cell mediated cytolysis. Okay. So CD16 also express CD16, which is involved in the antibody dependent cell mediated cytolysis, cytotoxicity. Now the another, that is CD16 allows NK cells to recognize and eliminate antibody coated target cell, that also help into the eliminate the target cell. Now the NK cell receptor, particularly activating and inhibitory receptor, is a main functional receptor. Okay, activating and inhibiting receptor. So inhibitory receptor, here it is the KIRS and NKG2A, and these all are the activating receptor. So first, KIRS and NKG2A, which binds to the MSC class 1 and block the NK cell activities. Okay, that means killing activity, which blocks the and activating molecules, or we can say activating receptor or marker that triggers the NK cell cytotoxicity if an inhibiting receptor is not occupied here. Okay, then and then only the activating receptor binds to these ligands, okay, MICA and B, and triggers the cytotoxic activity of the NK cell. Now both of these receptor group also fall into structure category: membrane with lectin-like extracellular region and membrane with the immunoglobulin extracellular region. Okay, they are both having the different extracellular region. Now the lectin, this is the protein which is particularly binds to the carbohydrate. Okay, however in this lectin-like receptor, okay, of NK cell, they binds to the protein rather than the carbohydrates. Now the extracellular structure of the NK cell receptor does not immediately identify it is inhibitory or activating receptor, right? So NK cell receptor is identically extracellular structure, can have different intracellular domain, okay, and therefore different signaling properties. For example, here it is the NK cell receptor, activating NK cell receptor. Okay, so ITAMs, here is the activating NK cell receptor. Okay, so and here it is the inhibitory NK cell receptor. Okay. So most of distinct phenotypic characteristic of NK cell, expression of set of unique activating and inhibitory NK cell receptor. Okay, Okay, so now that is the inhibitory NK cell receptor and ligands. So first, inhibitory receptor of nature killer cells are protein, as we know, and that binds to the MSC class 1 molecules on the surface of target cell or cancerous cell by the normal cells. Now the MSC1 molecules are like identification tags that healthy cells display to signal that they are normal and should not be targeted for the destruction. So in simple terms, we can say the decision of natural killer cells to kill or not is mainly influenced by the inhibitory receptor that attach to the MSC class 1 molecules. Right? When the inhibitory receptor on NK cell recognize the MSC class 1 molecules, and then they send the signals to the NK cell to prevent it from the attacking the healthy cells. Right, so let's discuss the in human which type of inhibitory receptor are present. So first, the most important receptor, that is the KIR, killer cell immunoglobulin like receptor. Here it is. Okay, are an example of inhibitory receptor of NK cell. Now another lectin like receptor, that is the functional LY49 receptor, are not present in human. So now unlike the primates, for example rodent like mice, use a different family of receptor called the lectin like receptor, that is LY49. They perform the same function to as KIRS, okay, which is inhibiting NK cell activity against the healthy cells by binding to the MSC class 1 molecules. Now here it's important note that functional LY49, as I said, receptor are not present in human, right? Now the both KIR and LY49, okay, so both are diverse and polymorphic. Polymorphic refers to the existence of multiple forms or variants, okay, of particular gene or traits within the population. Now the inhibitory receptors are generally specific for polymorphic region of MSC class 1. For example, KIR particularly binds to the particular MSC class 1, which is different from the normal MSC class 1. Okay, here we discuss like MSC class 1 molecules, okay, which is produced by the gene H2K and H2D in the mouse. Okay, so both are the H2K and H2D refers to different alleles of major histocompatibility complex class 1 molecules in mice. In mice, the H2 gene, there is a complex country gene that encode the MHC1, okay, molecules, and the H2K and H2DLS represent different genetic variants of MSC class 1 genes. Okay, so these alleles are associated with the variation amino acid sequence of the MSC class 1 molecules they encoded. There is a different amino acid sequencing. Now we discuss the human. Okay, in human, major histocompatibility complex class 1 molecules are encoded by the gene within the human leukocyte antigen, that is the HLA, HLA-B, HLA-C in the human, right? So this is how the particular receptor binds to the particular MSC class 1 molecules and disrupt the cell cytolysis, okay, or we can say prevent the cell cytolysis of the normal cells. Okay, so apart from the this inhibitory receptor is discussed in human and mouse. So human NK inhibitory receptor are Ig like molecules is found in the lectin like inhibitory receptor, that is the CD94 and NK G2A. Okay, this is the inhibitory receptor, this is the activating receptor. So in image we discuss and understand how the activation and inhibition occurs within the NK cells. Okay, so first NKG2A CD94, that is the inhibitory receptor. Here it is the NKG2C CD94, that is the activating receptor, right? Here it is the target cell, and both receptor present in NK cell and T cells. Now the first NKG2A CD94, okay, and another, that is the NKG2C CD94, signaling NK cell and T cells. Now it is the target cell expressing the HLE peptides can bind to the both NKG2A and NKG2C. Here it is there. Okay. The interaction of NKG2A CD94 receptor with the peptide presenting HLAE result in the phosphorylation of ITIM. Okay, phosphorylated ITIMs are responsible for recruiting and activating the intracellular phosphatase SHP1/2. Now, therefore, delivering the negative signals to the cells and give the signals to NK cell or T cell to inhibit the cell cytolysis here. Now, here it is the another way CD94 NKG2C heterodimer associated with the DEF12 contain immunoreceptor. Here it is the immune receptor tyrosine activating motifs ITAMS. Thus activating ZAP70 zeta chain associate protein kinase, that is the 70 kilodalton, and delivering activating signals. Okay, so now once the NK cell get the activating signals, they destroy the target cell. Now let's discuss the activating NK cell receptor and ligands. Okay, as we discussed the inhibitory receptor, it's time to discuss the activating NK cell receptor and ligands. So the most activating receptors expressed by the murine and human NK cell are structurally similar, and many are C-lectin like, so named because they have the calcium dependent carbohydrates. Okay, recognition domains. Okay. NKG2D, one of these activating receptor, is now recognized as one of the most activating receptor. Okay, so this is NKG2D are unique non-polymorphic MSC class 1 like molecule that don't binds with the beta microglobulin. And ligands, the NKG2D binds ligands are often induced on the cell expression stage, such as those with the DNA damage or infection. Okay, so now in LY49H activating receptors, okay, this receptor interact with the MSC class 1 like protein, which is not encoded by the host organism, but rather the produced by the murine virus known as the MCMV. Okay, that is the homology of the class 1. MCMV stands for murine cytomegalovirus. So in the absence of this receptor, okay, mice exhibit the poor protection against these common viruses. So this is different of activating receptor binds to their different ligands. Okay, so here it is the NK cell receptors. Some of the NKG2D species, mouse and human, structure lectin-like, ligand MSC class 1, activating, inhibiting, most but not activating. Okay, so example 2D activating by MIC-A, MIC-B. Okay, so these are the example of these receptors. These are the family, nature cytotoxic receptor family. Okay, they present in human, immunoglobulin in family. Now the how NK cell induce the apoptosis of their targets. Okay, so first in picture NK cell, here it is the lytic granules, target cell, signaling plus and minus. Okay, activating receptor, activating ligands, they present a target cell surface. Inhibitory receptor, okay, expressed by the NK cell, and inhibitory ligands, they expressed by the target cell. So in this first step, the recognition of target cell, first NK cell recognize the target cell, right? Now in this recognition, target cell assessing an expression of certain molecules on the cell surface. Okay, healthy cells are typically display normal levels of measured histocompatibility complex class 1 molecules. Now first they recognize of target cell, by the target cell produce the MSC class of molecules or not. Now second is lack of inhibitory signals. In normal circumstances, healthy cells express the MSC class of molecules that interact with the inhibitory receptor on the surface of the NK cells. These interactions send inhibitory signals to the NK cell and preventing them from the attacking healthy cells, right? As I discussed in previous slide. Now third, if the cells is stress or abnormality detection, So when target cell undergoes stress or either infection or become abnormal, such as cancerous, it may down-regulate MSC class 1 expression or express the stress-induced ligands. Okay, this reduces the inhibitory signals to NK cells and marks the target cell for potential attack. Now, for the activating signals, NK cell also possesses the activating receptor that recognize the stress-induced ligands or other activating signals on the surface of target cell. When these activating receptors engage with their ligands, they trigger a cascade of intercellular events that promote the activation of NK cells. Now, fifth, release of cytotoxic granules. When they get the signals, okay, activating receptor binds to the activating ligands which present in the target cell, and they gives the signals to NK cell. Then and then NK cell produce the cytotoxic granules. Okay, so activated NK cell release cytotoxic granules contain perforin granzyme. Okay, here you can see in this diagram, granules they release the perforin granzyme. As we know that perforin creates the pores, okay, in the target cell membrane, allowing the granzyme B to enter the cell. Now, sixth step is the granzyme mediated apoptosis. So once inside the target cells, granzyme activated cas pass, which are enzyme involved in the apoptotic pathways. Okay. Seventh, that is the FS-FSL interaction, okay, which is not shown in this diagram. Okay, which is also happen and also created by the NK cell that target cell apoptosis. Okay. So overall, NK cell use a combination of inhibitory and activating signals to discriminate between the healthy and abnormal cells. When target cells is identified as potential harmful, NK cell induce apoptosis through the release of cytotoxic granules and engagement of apoptosis pathways. So this is how NK cell induce the apoptosis of the target cell. Now here it is the MCQ based questions. First, okay, second, there are many MCQ based questions. Okay, solve these questions and provide the answer in the comment section below. Thank you for watching.